Hello, in this video we are going to talk about the diagnosis of respiratory viruses. Specifically, we will cover what respiratory viruses are, their consequences, current diagnostic tests, and introduce a new diagnostic technology known as DNA Nanobate. Meet Sarah. She just got back from the vacation of her dreams and is sneezing, has a stuffed nose, and a sore throat. Worried that this might not be the common cold, Sarah decides to call her doctor, who recommends she get a diagnostic test. After taking a diagnostic test, it is determined that Sarah has a respiratory virus. You might be wondering, what are respiratory viruses? Respiratory viruses cause infection of the nose, sinuses, or throat using a germ. Common symptoms of respiratory virus infection may include sneezing, sore throat, and a stuffed nose. While there are many different respiratory viruses, only 8 respiratory viruses have been identified by scientists as being capable of spreading from human to human. You may have heard of some of these viruses, as some common examples include influenza, coronavirus, and rhinovirus. Respiratory viruses have the ability to rapidly spread through communities and around the world. As shown with the recent pandemic, a small localized infection was able to rapidly spread around the world, resulting in a pandemic. Because these viruses spread quickly, it is equally important to be able to diagnose infected individuals in a timely manner. You might be wondering, how are these respiratory viruses diagnosed? The viruses can be distinguished from each other using a diagnostic test. The purpose of the diagnostic test is to determine which virus an individual has, and in some cases can also determine the specific variant of the virus. There are many different diagnostic tests that are used actively worldwide. However, some of the most popular tests include polymerase chain reaction, also known as PCR testing, and rapid antigen testing. These tests might sound familiar, as they are currently being used to detect positive cases of COVID-19 worldwide. You might wonder, why are new testing methods needed? In order to effectively treat a sick patient, it is important to rapidly and accurately identify the type and in some cases, the variant of the virus that the patient may have. Studies have shown that approximately 65% of respiratory viruses have been incorrectly diagnosed as pneumonia, and 95% of these misdiagnosed patients have received unnecessary medical treatment. This is because pneumonia shares many symptoms with other common respiratory viruses and cannot be differentiated from other respiratory viruses without advanced clinical testing, for example with a diagnostic test. As viruses spread, genomic mutations can occur, resulting in variants of respiratory viruses. Variants occur when there are small changes to the original virus. You can think of it like a tree branching outwards. Each branch is slightly different than the original branch it was connected to. Studies have shown that without proper, accurate, and quick diagnostic tests, respiratory viruses can wreak havoc on our healthcare systems and increase preventable deaths. From the pandemic, we have seen how current diagnostic testing methods have limitations, and new diagnostic testing methods must be explored, such as DNA Nanobate technology. You might be wondering, what is DNA Nanobate technology? DNA Nanobate technology uses a nanotechnology approach to detect multiple respiratory viruses and their variants at the same time. To understand how this technology works, it is important to understand what RNA is in a respiratory virus. RNA stands for ribonucleic acid and is present in all living cells. In addition, RNA is found in viruses. Viruses are not cells and are instead infectious microbes. Viruses infect and take over living cells in the host organism. Specifically, the genetic material of the virus is encoded in RNA, making detection difficult as human DNA produces RNA as well. When testing for the virus, the nucleic acid species are examined for the virus. When using DNA Nanobate technology, a method of biosensing called nanopore sensing is used to examine multiple different nucleic acid species. When current is passed through the DNA Nanobate, nanopore sensing is able to produce a graph showing spikes when viral RNA is not present and no spikes when the viral RNA is present. To use DNA Nanobate technology to detect multiple respiratory viruses and their variants at the same time, the following steps occur. The first step is that a nasopharyngeal sample is collected from the patient, which is taken by a swab through the nose to the upper throat. In step 2, the sample is cleaned to extract all nucleic acid. In step 3, a custom-designed enzyme known as RNase H is used to cut the desired target RNA out of the sample. In step 4, the DNA nanobate is prepared with captured DNA for the target RNA. In step 5, the isolated target RNA is captured by the DNA nanobate. In step 6, the DNA nanobate undergoes nanopore sensing, during which a current is applied and the results are recorded. A clinical trial titled Simultaneous Identification of Viruses and Viral Variants with Programmable DNA Nanobate by Filip Boskovic et al. Was run to test the effectiveness of using DNA nanobate technology as a novel diagnostic method. In this clinical trial, the presence of COVID-19 and its variants were examined. Swabs were collected from participants with COVID-19 symptoms, then the nucleic acid was extracted from these swabs, RT-PCR and nanobate testing were performed on the samples, and then the results were compared. In order to choose the target RNA for the DNA nanobate and nanopore sensing, researchers first had to examine the structure of the virus. Upon examining the structure, researchers identified 3 distinct targets, T1, T2, and T3, found in the virus. Using the identified targets, researchers tested the expected results from the DNA nanobate should the sample test positive for the virus. The clinical trial found that the speed of the diagnostic test can be significantly improved by using DNA nanobate technology. Specifically, using DNA nanobate, researchers were able to detect viral RNA in just 10 minutes. In addition, the DNA nanobate technology was able to show spikes when the targets were not present and no spikes when the targets were present. As seen in the graphs, the negative samples all had the spikes for T1, T2, and T3 present. However, when the sample was positive for the virus, the spikes for T1 or T2 or T3 were not present. Furthermore, to test the limits of the nanobate technology, researchers tested the technology to see if the diagnostic test could be used to accurately and successfully distinguish COVID-19 RNA from the COVID-19 variant RNA. The test results prove that the DNA nanobate technology could in fact be used for detecting not only virus variants, but numerous different viruses all at the same time. Further research is needed before DNA nanobate technology can be used clinically, as current research is limited. However, DNA nanobate technology is a promising upcoming diagnostic method for the identification of multiple respiratory viruses and their variants. Thanks for watching.
Created using Powtoon.